single-cell RNA sequencing has revolutionized our ability to study cellular heterogeneity at an unprecedented resolution. By measuring gene expression in individual cells, we can uncover rare cell types, trace developmental trajectories, and detect subtle transcriptional shifts that bulk sequencing simply cannot resolve. But there's a major catch. scRNA-seq data is notoriously sparse. A large fraction of the observed zeros in the expression matrix aren't actually biological absences, they're technical dropouts. These occur when transcripts fail to be captured during library preparation, even though they're truly present in the cell. And this sparsity distorts downstream analyses, from clustering and differential expression to trajectory inference, leading to false positives, blurred boundaries, and misleading biological conclusions. So how do we recover the true signal without fabricating expression? Enter imputation, the process of estimating missing values. Over the past decade, a wide array of methods have emerged. Some use probabilistic models, others rely on smoothing across neighborhoods, and more recent approaches leverage deep learning to capture complex nonlinear patterns. But many of these methods struggle with a fundamental trade-off. They either oversmooth the data, blurring cell type distinctions, or they impute too aggressively, inflating true zeros and creating artificial signals. What if we could separate the detection of dropouts from their imputation, treating each step with the right tools? That's the core idea behind SCR-MF, a modular two-stage framework that combines scRecover for principal dropout detection with missForest for robust, non-parametric imputation. Let's break it down. First, scRecover identifies which zeros are likely technical dropouts. It does this using a zero-inflated negative binomial, ZINB, model, a statistical framework that explicitly models two sources of zeros, biological silence and technical dropout. For each gene, it estimates the probability that an observed zero is a dropout rather than a true absence. This is done via an expectation maximization algorithm that iteratively refines parameter estimates for gene-specific dispersion and capture efficiency. But here's the key insight. Instead of imputing all zeros, scRecover uses a species-accumulation style extrapolation to estimate how many zeros per cell are likely due to dropout, not which specific ones. This avoids flagging too many entries in already sparse cells. The result? A binary mask, a map of where imputation should happen. Zeros flagged as potential dropouts get marked for recovery. All others, including genuine biological zeros, are preserved. Now for the second stage, missForest. This is where the real power comes in. missForest is a non-parametric, iterative imputation algorithm built on random forests. It doesn't assume any specific distribution for the data, no normality, no linearity. Instead, it learns complex, high-order interactions between genes through decision trees. And crucially, it only imputes the entries flagged in the mask, never touching the biological zeros. The process works like this. Start with a copy of the original matrix, filling masked entries with simple column statistics. Then, iteratively, for each gene with missing values, train a random forest using all other genes as predictors, and use it to predict the missing entries. Repeat this process until convergence, monitored via out-of-bag error or a normalized difference between iterations. Because random forests naturally handle mixed data types, capture nonlinear dependencies, and provide built-in error estimates, they're ideal for scRNA-seq, where gene expression relationships are rarely linear or additive. And because imputation is only applied to the flagged entries, not all zeros, SCR-MF avoids the common pitfall of oversmoothing. It preserves rare cell states, maintains heterogeneity, and prevents the artificial tightening of clusters that plagues many other methods. We tested scR-MF across multiple real-world and simulated datasets. On the GSE 86982 dataset, a time-resolved neural differentiation study of over 1,800 human stem cells, scR-MF achieved an adjusted RAND index, ARI of 0.82, and normalized mutual information, NMI of 0.88. That's significantly better than MAGIC, ARI 0.68, Viper, ARI 0.71, scImpute, ARI 0.79, and even DeepImpute, ARI 0.76. On GSE 75748, a developmental lineage dataset, scR-MF again led the pack with ARI 0.77 and NMI 0.81, outperforming all baselines except Scrabble, but only when bulk RNA-seq data was available. In simulated datasets with up to 80% dropout, scR-MF gained 9 to 12% relative improvement in ARI over the next best method. But numbers alone don't tell the full story. Visually, the clusters became tighter and more distinct after imputation. The global mean expression increased, as expected, from 237 to 256, while the confidence interval widened slightly, reflecting improved signal-to-noise ratio. Most importantly, known marker genes like SOX2 and PAX6 showed dramatically higher expression coherence in neural lineages, a clear sign of improved biological fidelity. And unlike methods like MAGIC or Viper, which often merge nearby subtypes due to aggressive smoothing, scR-MF preserved fine-grained differences, critical for studying lineage bifurcations or transitional states. Now let's talk about trade-offs. The main limitation: computational cost. Random forest on large, sparse matrices are resource-intensive. But we mitigated this by using small forests, just 10 trees, and limiting imputation passes to 2 iterations. This reduced runtime dramatically, while retaining 95% of the accuracy. For faster execution, we also tested missRanger, a more efficient variant of missForest. It delivered broadly comparable performance with faster runtimes, offering a practical alternative when time is tight. And here's a critical point: modularity is key. SCR-MF isn't tied to one specific detector or imputer. You could swap scRecover for scImpute, or missForest for a deep learning model. Though in our experiments, the combination of scRecover's precise dropout detection with missForest's flexible regression proved most effective, especially on heterogeneous datasets. So why does this matter? Because imputation isn't just about filling gaps, it's about preserving biological truth. When you impute indiscriminately, you risk turning silence into signal, rare states into noise, and natural variation into artifacts. scR-MF gives you control. It detects dropouts with statistical rigor, imputes only what's needed, and leaves the rest untouched. This makes it not just more accurate, but more interpretable, a crucial advantage in scientific discovery. Looking ahead, future work will focus on scalability, integrating GPU acceleration, sketching techniques, and early stopping to speed up computation. We're also exploring tighter coupling of detection and imputation within a unified probabilistic framework, and combining scR-MF with deep generative models like SCVI or DCA to capture nonlinear manifolds more effectively. But for now, scR-MF stands as a powerful, principled solution, a hybrid computational intelligence framework that balances accuracy, transparency, and biological fidelity. It's not the end of the road, but it's a significant step forward in unlocking the full potential of single-cell transcriptomics. Because in the world of single-cell data, the real challenge isn't just measuring expression, it's knowing what to believe. And scR-MF helps us believe in the signal, without losing sight of the silence.